we're looking at, and then I will go into the more specifics. So you can imagine the human genome as a library with books. It's actually this printed library of books of the human genome in very very tiny print. And the genome consists of this of messages that total about 3 billion letters. Each person, each one of us, has 2 copies of this library. So each book, let's say each chromosome, exists in our cells, and each and every one of our cells has 2 copies, one that comes from our father and one that comes from our mother. Another interesting aspect of these books is that some parts of the books should be read left to right, and other parts should be read right to left. So there is essentially, if you think about it, there's about 6 billion letters, because it's in both orientations, that contain information that is sufficient to build a human, and keep a human being going through life. So the organization of these of the genome is primarily through some functional entities called genes. We don't need to go too much into detail, because what is a gene is a long and complicated discussion. But for our purposes, you can understand the gene as a part of the genome, a location on the genome that encodes a specific function. So genes are still made out of sequence, out of letters, letters corresponding to chemical molecules. And importantly, these genes encode messages in a molecule called RNA, and then this RNA molecule can go and do things, can have function into itself, or it can be an intermediate to produce other biological molecules called proteins. RNAs and proteins are the structural and functional machinery of the cell, meaning they are what the cell is primarily built of and what does different operations. Out of all these 3 billion or 6 billion, however you want to measure it, letters in a human genome, only approximately 1% encodes for genes. The rest has either regulatory functions, meaning it regulates how much of a gene is going to produce messages in this specific cell, or it doesn't encode anything, the famous junk DNA, or it could have other functions such as spacing and things like that. Mostly, in most cases, we generally tend to focus on the parts of the genome that have genes and the regulatory regions that are usually nearby, more for reasons of simplicity than anything else. Okay. So each person, each and every one of us, has a different genome. By and large, our genomes are similar, but they will have variation. There will be variation. So out of these 3 billion letters in a single copy of the genome, and as I said, each one of us has 2 copies of the genome, one from the mother and one from the father. Each person has about 1,000 to 2,000 letters that are different compared to the standard, let's say, consensus genome. So, of course, what we said also before is that only 1% of the genome encodes genes, so most variants make no real difference in how our bodies work, let's say, what cells do. However, that said, there are some variants that could have very, very important effects. For example, in things we call monogenic traits, and I will come back to what that means, a single variation in one letter of the genome, one specific letter, in one specific location, could affect things drastically. So, for example, in the disease called sickle cell anemia, a single variation in one letter of the gene that includes hemoglobin, that's a protein that's found in red blood cells, it can produce red blood cells that take this characteristic shape, and they're not as efficient in carrying oxygen through the bloodstream, leading to disease. So monogenic means a single gene is affected. A single gene being affected is enough to lead to this disease. These are generally easier traits to study because it's essentially you don't need to worry about the whole genome. You can only focus on the one gene. We're not going to talk about these traits today. We're going to talk about the opposite, polygenic traits. So polygenic traits mean that multiple genes combining in various biological pathways that are very hard usually to follow, feed into some control networks, and in the end give a result that could be a disease, or it could be a trait like height, for example, or weight, or something else that has a genetic component. And these are quite hard to study and narrow down. And actually, in many cases, it takes difficult and kind of very, very complex experiments in order to even prove that these traits, for example, I mean, height is more easy, but other traits have a genetic component, usually by looking at twin studies, so looking at people that were twins separated at birth, and if they tend to have closer the traits correlate more than expected. So what we are going to look at today is how these polygenic traits can be, like, basically we are trying to predict the risk or the chance that an individual will develop one of these polygenic traits, for example, heart disease or Alzheimer's disease during their lifetime, based on their genome, on these variations that exist on their genes. There are quite a few problems. The one is that a single trait or disease could come from many different pathways. For example, there is not one cancer, or even one specific, let's say, skin cancer. Someone could get skin cancer through multiple different pathways. Same with heart disease, same with other diseases. There are many mechanisms that we don't know about. So We are trying to discover what this mechanism could be. And the effects could happen over a long period of time in various cells and tissues, and they're very hard to model and very hard to monitor. What we have working for us is that pretty much all the cells in a human body for a specific individual have the exact same copy of the genome. So we know the one thing that is mostly, that's an oversimplification, but mostly written in stone, is that every cell in our body has the same genome. So looking at variation at the genome level allows us to make predictions for any, let's say, stage of our life, and any type of any organ, any tissue in our body, knowing that the context in which everything will be working in that tissue or in that organ will be the specific genome that the organism, the individual has. So we have some tools in biology to actually look at these letters, and here they're listed three levels, from more expensive and more thorough to the least expensive, easier to do, and lower information. Whole genome sequencing means we can take a sample from a person and we can read every single letter in their genome. Then we get a big file, a few gigabytes, that has, for every position, what is the letter that individual had in the two copies of their genome. Exome sequence is pretty much the same, but it's only in the parts of the genome that encode for known genes. So we cut down the 99% of the genome that doesn't really encode for genes, and we focus on the parts that are more important. Of course, this comes with, you might be missing some parts of the genome that could be doing some that we don't know about, but also you are reducing the cost and making things, making files smaller and so on. And in the finally, you can have specific panels that read specific letters. So we know, for example, that maybe at position, I don't know, chromosome 3, position 1000, whatever, this letter, this specific letter, if it's flipped, it could create a big problem, and we want to specifically measure that. So you can build these panels where you can read specific few thousand specific positions, and then use that for whatever you want to use it for. This is what what are they called? This 23andMe or this ancestry companies use. They only measure specific things that they know they reflect traits that they want to talk about. So once we have experimentally, let's say, measured, experimentally measured the letters, then we, at each position of the genome, we can go and we can look at the reference genome, which the reference genome is, let's say, a consensus genome that has been created by several, by measuring several individuals. And we can go and we can look at that exact position. Is the person we are genotyping a homozygote, which means they have the same letter from both parents? Are they a heterozygote, which means they have two different letters from the parents? Or are they homozygote alt, meaning they have the same letter from both parents, but it's different from the reference genome? And this is, as you understand, is important because then we can we can identify parts of the genome where each person has variants, and this can be very important for identifying these traits in the disease. So when you have for example, two genes here, I've made like, let's say, two locations on the genome that encode for two genes, and you can see in the two copies, for example, of the genome that this individual has, I put with a black line the positions where they have a variant compared to the consensus genome. And you can see that in some places, these two I have identified, they actually have, from both parents, they have the variant letter. Usually, what happens is that, let's say where gene A is, if they had, let's say they didn't have any homozygous alt, then the copies of this gene coming from the, let's say, maybe in the copy coming from the father, for example, there could be a problem, and that gene was not being produced correctly. But then the other copy would compensate for that. But if there is a problem on both sides, then this can be much more detrimental. So that's why we want to know if genotyping is important, right? If there's a change on both sides or just one. Generally, what we have is we have a bunch of individuals, a large number of individuals. We know who have, for example, a disease and who don't. So we have case and controls. And we also have the genotypes. So we have the genome read, or maybe the exon, but we have a number of variants for each one of these individuals where their genome has a different letter than the standard genome. And the main task that we're talking about here is, given these people, these samples, and all their variants, we want to produce a score that can tell us the probability or the risk that each individual has of having or not, the probability that they basically will have or develop this disease or have this trait. So very roughly speaking, if I have, let's say, 1,000 individuals with that developed, I don't know, a specific disease, and 1,000 individuals that didn't develop this disease, and I can find the variants that can tell one group from the other, then potentially when, let's say, a baby is born, I could take the genome, I could look at these variants, and I could say, there's this much, you have this much chance of developing this disease in your life. So, maybe specific interventions need to happen, or it's very useful for medical characteristics. The issues, of course, are that measuring the genotypes is expensive, difficult to standardize, there's large variability, every study is done slightly differently, so some data sets are hard to get to and hard to standardize. There are many variants, so there are hundreds of thousands of variants that have some kind of effect, and there are millions of variants just in total. And usually you have in the hundreds, or if you're like in the few thousands of samples. So that can be, the dimensions are difficult for machine learning if you have few samples and many variants. And then the other issue is that you can have, you could have many people that, for example, have a heart attack, but maybe many of them or some of them will have it for a different reason. Maybe it's a different biological pathway that leads to that. So this will be subclasses, subgroups of your disease class that will show completely different profiles. And when you try to figure something out that is statistically significant, maybe this will not be possible because of how they have different backgrounds. Okay, so this is the introduction about what we're looking at, and now how the ways that people have up to now kind of approach this and try to solve it. So the main, let's say more traditional standard way to approach this is called the polygenic risk score. So a polygenic risk score is an estimate of an individual's genetic liability to a trait or disease. So you're trying to estimate what is the chance that this individual has or doesn't have that disease of the genetic aspect, right? And it is calculated, it needs to be, it is traditionally calculated using two things. The individual's genotype, so the letters that we have measured, and a background that comes from a big study where many thousands, ideally, or tens of thousands or more of individuals have been genotyped, and each variant has been associated with the disease or with the trait with a probability. So these genome-wide association studies, essentially, what they do is they will take, for example, cases and controls, and they will look at each position on the genome when there's a variant. What is the probability? What is the basically how often do cases have this variant versus how often do controls have this variant? And they will assign a p-value for that. So this, I don't know if you can see it very well, but this is a graph where on the x-axis there is every single position of the chromosome, and the y-axis is the minus log p-value. So the higher this is, the more associated this variant is with the cases versus the controls. And then what normally will happen is there will be some steps, which I will explain right after, where basically you go from this p-value, this association of this correlation essentially between being a case and having this variant to a predictive score. I'm not going to go too deep into this, but there's a very nice review paper on genome-wide association studies that you can look at and explain. It explains pretty much everything. There's also a GWAS catalog where people have publicly published their studies. So there's over 60,000 studies for various diseases and traits and so on. So these are publicly available data that you can get. What's important to say here is that it doesn't mean that if there's a genome-wide association study, for example, with 1,000 individuals that finds that looks at the connection between a trait and the genotype, it doesn't mean that they will give the genomes of every single individual, probably not. But what they will give is a report of variants and their associations of their p-value compared to that disease. Okay, so now going from this GWAS to the polygenic risk score. So basically, So basically, what you are going for here, what people are going for here, is basically you're trying to take this p-value that has been designated for each single letter in the genome or in part of the genome, and you're trying to combine these values into a specific score that can then be used to give a relative, let's say, to give a, like, it's not usually an absolute score that will tell you, I don't know, in how many years, for example, someone is going to get this disease, but it will be a score that, the higher the score, the more chance someone has to get the disease, something around that level. So this is also a very nice review article explaining everything about polygenic risk scores in the traditional sense. And I will go very, very quickly. I will not go through how the data, I already explained kind of how the data is collected. I will not go through quality control because it's very technical, but I will go over a little bit the calculations, and so maybe you will see some things about why the other techniques that I will show later also make sense. So the first thing is the linkage disequilibrium adjustment. So this is a big thing in genetics. Basically, what happens is we have, if you think of all these variants, you could think of them as variables, right? So an individual is, let's say, if there's a function that takes into account all these variants and then gives, the outcome. But what happens in reality, because of how replication of DNA works and so on, is that variants that are close to each other are not independent from each other, close in space from each other on the genome, are not independent from each other. So they tend to be to have they tend to stay together, and this can create a problem if you so let's say if there's one variant that actually does something, this will have a very high p-value, for example, minus log 10, very high p-value, but also variants next to it will also have inflated p-values just because they're close by to the one that actually does the thing. And that is called linkage disequilibrium. And it's a big problem because in polygenic risk scores, generally what people want to do in the end is you want to get a score per variant, and then you want to sum them up to give the final risk score. So if you take every variant that goes above a certain threshold, you're going to end up with many variants, many variables essentially that are giving you information about the same thing, and you don't want that. So there are many ways to let's say avoid this. The most common one is called clumping, and basically what that means is that you define how closely correlated the variants will be to consider them as in this equilibrium, and then you pick the one with the best p-value and remove the rest. So now let's say you have your selected variants. Now your number of variants is much smaller. You could have, of course, still in the thousands. And then generally what people do in this polygenic risk score estimation is they use some kind of mathematical operation or statistical operation, I don't know how to say it, to scale the scores, to shrink the scores, so that the extreme values, extremes of the p-value don't give as much weight as the bulk of the scores. And this, from what I mean, I've read in several papers, that is known to increase predictive power, but how exactly, I'm not sure. And then after that, or along with that, not every variant selected. There will be a threshold in p-values, and depending how strict that threshold is, less variants will be considered in the model, and this will, of course, make it easier to test, and so on. So they're prioritizing basically variants that are more relevant to the question. So combining all these things together, the polygenic risk score is calculated and or computed, usually as a sum or a multiplication of different scores, and then it is tested. So there are usually two data sets. The one is the genome-wide association study, that is a big study with thousands of participants on. And then usually there is another smaller data set here called the target data, which could be just a few individuals maybe of a more focused population or something. And this is what we're actually trying to predict. So so basically, the findings from the big GWAS data set are then applied onto the smaller target data set, and then things like regression are calculated to see how well that polygenic risk score performs. You will generally see graphs something like that. So, for example, here, the first graph, you can see this is a calculation of BMI. This is a kind of weight by height, let's say. And you can see here, for example, that these are the predictions. So this is the polygenic risk score for BMI in quantiles. And then you can see the actual measurements in the target population and how it shows basically that the values go up as the score goes up, which means there is some, let's say, it's a good score because it goes it goes up. If you here is similar, it's something similar, only these are equally divided. Here you get 1 point for 99 to 100, then the top 5%, or the next 5%, and so on. Same here, and you can see the actual values. So you can see that yes, things are generally sorted, but for example, there could be an individual here that is predicted by score to be at the top 1% of weight, and they are actually in one of the lowest, in actual BMI, they are in one of the lowest values. So we are very far away from having good polygenic risk scores for very complex traits. Okay, I have another maybe 10 minutes, so now I will go into the other approaches that recently are being shown to outperform the polygenic risk scores and are based more on machine learning. One of the big ones is called lasso regression, and this is a similar approach to Reed's regression, and it's basically the important thing to understand for this is that it can do two things at the same time, which is quite interesting for us. Is that it can estimate a value, but it can also remove variables that don't give information to reach that score. So basically, you can estimate, for example, if you're trying to predict BMI, you can estimate the BMI using this model, but it will also remove the variants that don't play into this estimation. So this is very good because it makes it makes the model more explainable. So you can, in the medical field, it's very important to be able to explain why you're making a prediction, and this allows, for this explainability and reduction of variables to be tested, and so on. There is one. I'm sure some of you know probably the maths better than me, but there is a variable or a parameter called lambda in Lasso regression, which is, it has to do with, roughly speaking, it has to do with how much weight the actual values will get versus the training of, I don't know how to explain it very well, the values versus the model. In a way, it's hard to explain if I don't explain everything about how Lasso works. But how to get the proper lambda value is a big question, and there are papers talking about that, and it's like kind of an active research about how to get a lambda that makes sense, that leads to better predictions. So Lasso is quite established. It's quite an established method. It's I think like the linear polygenic risk scores, Lasso are the most commonly used things, and then on top of that, other people now are trying different ideas. The other one I found quite interesting is from 2 years ago, so it's quite recent. All of these are quite recent, actually. And this is using essentially Lasso as a Lasso to prioritize variables or variants, and then it's using XGBoost, which is a tree-based method, to actually do the classification. And that they show here, for example, in the prediction of height as a phenotype, but they have many. They've done it on many traits. They show that for example, if you use the polygenic risk score, the original stuff I was talking about, you would have about 1 million parameters. With Lasso, this you can reduce that to 4,800, and then you can train the XGBoost on that, and then combine that with the polygenic risk scores to get even better accuracy with a much smaller number of final parameters, which is quite nice. So this is one solution that seems to be quite promising. Another one, and I think it's the last, let's say, thing I have, is now people are using, of course, neural networks, as with many things these days, and they actually are using neural networks to predict, predict polygenic diseases. And this is a quite complex flow chart of how this method works. One thing I completely glossed over is that what plays a lot into this is also that different populations have different variants, and sometimes the case that if the big genome-wide association study you're working with was done, for example, in Europeans, but then you want to predict in Asians, for example, you might be not as accurate because your data doesn't come from that background. So many models are trying to use this kind of mixture of big data sets to combine them together. Anyway, that's kind of an aside. We can maybe have another talk about that. But anyway, so they use lasso and neural networks, and they predict. They produce a score, and they saw in their paper, which it's a nice paper, that in two different data sets, the red line is the neural network. It works better than the lasso, and quite better than the polygenic risk-score, the linear polygenic risk-score, in being predictive about these disease, Alzheimer's disease. Okay, so this was pretty much everything. I could explain a quite complex, field in an hour. So what is going on now? What's coming is that we are looking at a lot more data. Millions and millions and millions of genomes are going to, are currently being sequenced. Sequencing becoming cheaper. We are also looking at many more isolated small data sets that various, like now it's within grasp of normal researchers to do, 50 to 100 genomes. It's not like crazy to think about anymore, which, maybe 10 or 20 years ago was, something a big consortium would do. So we started getting these small data sets that somehow need, they could have quite interesting, they could be quite interesting in predicting things, but they are isolated, and often because of regulations and legal difficulties, it's not very easy to share the data. And of course, all of this, I didn't focus too much on the applications, but having this predictive power could open great potential for medical breakthroughs. So thank you for hopefully following my talk, and we can discuss if you want any questions or comments or whatever. Thank you.
Thank you, Fanus. That was very insightful. I will invite people, if you want to type your question on the chat, or you are welcome to join the audience if you want to ask any question or any make any point, please. No questions? Come on. If not, I will be forced to come up with a question myself.
It's okay. It's late.
Yeah. I know it's afternoon, so don't worry. Or maybe I should, because it's recording, maybe I should stop recording.
Yeah.
That will make people feel more comfortable. Let me do that to see. If not, I have some questions.